This is the pre-lab talk for experiment number 8 for Chem 120, the acid-base titration. The goals for this experiment are to determine the acid ionization constant, or Ka, for acetic acid using a titration. We will also get a chance to observe the pH behavior during our weak acid strong base titration by looking at the general shape of our pH curve and taking a closer look at the buffer region. We will look at how the pH changes in the buffer region by using the acid-base indicator bromocresol green, or BCG, during our titration. We will also assess the quality of our data using the standard deviation of the mean. Today's safety pause reminds us that we'll be working with mildly corrosive acids and bases at the concentrations used in this experiment. We want to make sure that we wash our hands before leaving lab and wear gloves when necessary. We also want to remind you that acids need to be collected and neutralized before being flushed down the drain with plenty of water as disposal. Another reminder that might not have been mentioned before is that when we wear our lab coat and goggles in lab, we do need to make sure we remove these pieces of PPE, particularly the lab coat and any gloves, before leaving the laboratory, so that we do not contaminate other areas with chemicals. Let's take a look at the reactions involved in this experiment. The first reaction we want to take a look at is the titration reaction itself, which is an acid-base neutralization, where we react acetic acid with sodium hydroxide to form sodium acetate and H2O. This reaction is highly favorable and is driven forward by its spontaneity and high equilibrium constant. Typically, when we run this lab in person, we assign students a particular molarity of NaOH and ask them to deliver 50.00 milliliters of that solution to their Erlenmeyer flask for the titration. We will be delivering the 50 milliliters using a 50 milliliter volumetric pipette. We will then add to that solution from our burette about 0.2 molar solution of NaOH. The goal of this experiment is to see how much sodium hydroxide it takes to reach the equivalence point of the titration. This can be calculated as a first prediction if you have this concentration of acetic acid. You'll be asked to do this in your lab assignment. You can then estimate the volume of NaOH added to see if it is consistent with your calculation. However, the majority of this lab is looking at the acid ionization equilibrium, which is shown here. When we write an acid ionization equilibrium, we show the reaction between our acid, in this case acetic acid, and water. The proton is transferred from acetic acid to water to form H3O plus and the acetate ion. We can write the Ka expression to show that Ka does depend on the hydronium ion concentration and the ratio between the concentration of acetate and the concentration of acetic acid. We mentioned in our goals that we want to determine Ka. In this experiment, we'll use our titration data to determine the Ka by measuring the pH of our solution at different points during the titration, and by using a color matching color matching procedure with some reference solutions determine the ratio between acetate and acetic acid in the buffer region of our titration. Therefore, in this experiment, we can use several points during the titration to calculate values for acetic acid, values for the Ka of acetic acid, which should be the same, right? Let's take a look. So let's take a look at our reference solutions. First, let's talk about bromocresol green. Most acid-base indicators themselves are weak acids, as discussed in experiment 5. Therefore, the protonated and deprotonated forms of bromocresol green are both present in the solution. The color that the solution appears depends on the ratio between the deprotonated form and the protonated form of bromocresol green. Typically, for an acid-base indicator, an acid-base indicator changes color around the pKa of the indicator itself. For bromocresol green, this color change ratio is between around pH 4 and pH 6. If you look at the figure of the titration curve, you see that this color change range coincides with the buffer region of acetic acid. Therefore, we can use our color matching to figure out the ratio of acetate and acetic acid. You can see in the picture that 7 reference solutions will be prepared, ranging from having no acetate, which is solution A, to having no acetic acid, which is solution G, and everything in between. In these different reference solutions with certain ratios of sodium acetate to acetic acid, we will add bromocresol green to get our different colors. These solutions are typically prepared by using a burette so that we get very precise volumes and amounts of sodium acetate and acetic acid in our solutions. Let's take a look at the procedural points to outline how we're going to do this. To do our color matching, we will as we add our NaOH to our acetic acid solution, which will also contain bromocresol green, we'll have to bring out our reference solutions to see how the color matches up with each solution. Sometimes they'll match up completely with one of the solutions, but oftentimes they'll be somewhere in between. We will use a 4-letter designation to determine the color. For example, at the start of our titration, where there's just acetic acid, our titration solution will look a lot like reference solution A. We might call this AAAA. 4 A's to represent that the solution looks just like reference solution A. We can also get to a point where the titration solution looks somewhere right in between reference solution A and reference solution B. We can therefore call it AABB. Finally, if we have a solution that looks almost like solution B, but not quite there, we could call it ABBB. You'll see us do this color matching during the lab video. Another important point that you'll notice about the video is that it's quite long. We have to do this titration slowly to get the best color matching for our determination of Ka. We'll especially titrate pretty slowly in the buffer region and definitely slow down a lot during the equivalence point of the titration so that you can get a good estimate of that equivalence point volume. Another important thing to note is that you'll notice that we're using a special logger profile in the lab video for this titration. This logger profile has has a third column for our reference solution matching and already sets up events with entry. Remember that events with entry allows us to input a specific volume, a specific amount, a specific volume at our measured pH. We will still need to calibrate the pH sensor, however, before starting the titration.